Hello guys, today we are going to discuss homeotic genes, its role in development of an organism. But in this, I will make it very clear that what are the homeotic genes and what the different tutorials are providing the information. The difference I am going to discuss. So here the definitions is like relating to caused by or being a gene producing a usually major shift in developmental fate of an organ or body part. And one more definition is that these genes involved in development and differentiation, controlling the formation of whole organism or body parts in animals and plants. So both the definitions are the same in their explanation. Here in the first definition, it is a comprehensive form of the definition. In the second definition, it is a little expanded form of the explanation. So here it is the development of an organ or body part. Here it is involved in development and differentiation. So here development and differentiation are the one which is controlled by these homeotic genes when the organism is going to develop it. Either it can be in animals or in plants. So coming to the next slide, during development of an organism, these homeotic genes are going to act as master regulator genes, where these are going to unfold the genetic developmental program of body plan in various organisms, particularly the body segments or structures. Now here, unfolding of genetic developmental program of body plan in various organisms. So here commonly in all organisms there will be homeotic genes which will be responsible for the unfolding of the development. This unfolding of the development will take place after the blastula stage, that is in the late blastula stage, which is going to recognize the formation of the gastrulation layer. During this time there will be the particular body segment or structures. Body segments in the sense, if you can see, the insects or in some organisms during the early embryonic stages there will be the body segments. In some organisms there will be the structures. So depending upon body segments or the body structures, those are going to develop into the further stages of the organisms. So here these are going to regulate in the sense they are going to send the signal to the further gene networks which are responsible for the further development in the downstream. So here it is very much clear that there will be the transcription factor proteins which inducts the signal to downstream cascade gene networks involved in body patterning. So here these are going to act as the master regulator or the control genes. These are going to signal to the downstream cascade gene network which is going to further take forward the developmental process. Each homeotic gene is regulated from subset of genes of homeobox. Here each homeotic gene is nothing but there will be the cluster of genes which are present in the homeobox. This homeobox is going to lodge many number of the clusters of the homeotic genes within it. So this is what the meaning of this sentence. The homeobox is the conserved DNA sequence of homeodomain. It is, as we have said or discussed, that these homeotic genes is going to present in the form of the subsets of genes in the homeobox. As we know that gene is a genetic material, is of the DNA, but it is conserved DNA sequence of homeodomain. Homeodomain in the sense, homeobox is conserved DNA sequence of homeodomain. domain. It is the homeodomain which is going to express the transcription factor. So we are going to discuss this homeobox and homeodomain further more in the coming slides. So homeotic genes are found in many animals of echinoderms, insects, mammals and plants. So here it is going to cover the major part of the kingdom Animalia. When you are going to consider in all multicellular organisms or metazoans, these homeotic genes are responsible for the development of the organisms from its embryonic stage. So further in the next slide, mutations caused in homeotic genes cause displaced body parts, such phenomenon called as the homeosis. It is no need to explain in the detail, as you know that there are some examples of the homeotic gene studies in Drosophila melanogaster during its early embryonic stage, and also in Arabidopsis thaliana. There you are going to induce the mutations at certain parts of the developing embryo, and you are going to come across the results where you are going to see the abnormal or mutated or aberrant organism developed. Whatever the induced mutations you have done to the early embryonic stages of these organisms, those are the homeotic genes which got mutated and they have given the different results. That is the thing. Causes displaced body parts. Such phenomenon is called homeosis. Well, for this, a simple example is of the Drosophila antennapedia. Now here you are going to mutate the antenna developing homeotic gene. That is what the cascade of the signals that has to pass till to the complete organism development. At the initial stage itself, you are going to mutate or induce the mutation. Because of that, at the place of the antenna, the leg is going to develop. That is what the displaced body parts, such phenomena is called a homeosis. And further, mutations that lead to development of structures in an abnormal place called ectopic structures of body are usually lethal. Certainly, as we have discussed here, that is the displaced body parts. But here, at some positions, the mutations causes the are going to lead to the lethal situation to the organisms. That is called to be as the ectopic structures. Now further, in this slide, we are going to discuss the homeobox. A homeobox is a DNA sequence. This homeobox are of 180 base pairs long DNA sequence. A homeobox found within genes that are involved in the regulation of the patterns of anatomical development. And a homeobox anatomical development is observed in animals, fungi, plants and numerous single cell eukaryotes. When you are going to consider the first and second point, you can understand it is a very simple. It is the very much conserved DNA sequence and they are going to have 180 base pairs. Whereas a homeobox found within genes that are involved in regulation of patterns of anatomical development. Here, put together all the homeotic genes clusters has been explained. So all is going to responsible for the regulation of the patterns of anatomical development or the morphogenesis. You can understand what is anatomical development also and what is morphogenesis. Both are the same, that is the development of an organism from its embryonic stage. Now this particular homeoboxes has been observed in the animals, fungi, plants and numerous single cell eukaryotes. Now coming to the homeodomain, I have made this slide to be the homeobox. Here I have prepared this slide with Homeobox and Homeodomain. Remember, Homeobox is going to lodge the homeodomain. So we will discuss here what is homeodomain. Homeobox genes encode homeodomain protein products that are transcription factors. What is homeodomain? Homeodomain is a particular expressing DNA sequence which will be identified and it will be transcripted. Further, these homeodomain protein products that are going to act as the transcription factors. So these will be synthesized as a characteristic protein fold structure that binds DNA to regulate expression of the target genes. Now you can understand that the further cascade of the expression of the target genes has to takes place. So for that reason, these homeodomains within the homeobox is going to get expressed, and further they are going to regulate the target genes. Homeodomain proteins regulate early embryonic gene expression and cell differentiation during development. Now you can understand that these homeodomains are targeting those genes which are responsible to early embryonic gene expressions and cell differentiation during development. So during this time, if you are going to induce mutations in homeobox genes, can cause developmental disorders. Now here you can see this line is very much particular to make you to understand within this general explanation that if you are going to induce the mutation, that mutations are going to cause the abnormal expressions in the developed organism. So during that completely developed organism is going to show any abnormality or aberrant characteristics that is going to make us to understand what the gene got mutated. So here this is how the homeodomain is going to be present. Now this will be the structure of the homeodomain in the homeobox genes. Now you could have understood what are homeotic genes and how they are present in the homeobox and what is the homeodomain. These three are the three different understandings, but all these three are togetherly present. So you can understand this now in this particular smart graphics which I have tried to prepare, and I hope you people will understand. Now here homeotic genes, we are discussing the topic is the homeotic genes. Now these homeotic genes are present in the homeobox in the form of the different clusters. Now this homeobox will be having the homeodomain. This homeodomain is going to be present in the form of the four main different clusters of the homeotic genes. One is the HOX, next is the LIM, next is the PAX, and the next is the POW. So these are the four different homeotic gene clusters which are present in the animal kingdom. These four different homeotic gene clusters will be present in the homeobox and expresses through the homeodomain protein transcription factors. You can understand that homeotic genes are nothing but they are present in the homeobox and they are going to express through the homeodomain protein transcription factors. Further, it will be expressed as the HOX genes, Ilium genes, PAX genes, and POW genes. Now, under this, there are several subset of homeotic genes. So as we have discussed, these are the several subset of homeotic genes present in the homeobox. In these subsets of homeotic genes, Hox genes are one larger subset, and next is the Ilium genes, PAG genes, POW genes. Now, Hox genes are a subset of homeobox genes. Hox genes are essential to determine the identity of embryonic regions along the anterior-posterior axis. Hox genes are important metazoan genes during embryonic development. Hox genes has this phenomenon called collinearity, where the linear order of Hox genes within a cluster is directly correlated to the order they are expressed in both time and space during development. Here, the first and the second sentence of being the subset of homeobox genes and will be responsible to determine the anterior posterior axis during the embryonic development or the simple lines. Here, the third point is important in the metazoan genes during embryonic development. Metazoans is nothing but all the multicellular organisms which are well developed with the organs. So those are the metazoans. So within these metazoans, these Hox genes will be responsible for the complete embryonic development. So that is the meaning of this sentence. And further, it is collinearity. Collinearity in the sense, these Hox genes, when they are going to express in the homeobox through the homeodomain, these cluster of Hox genes will be responsible to correlate each out within this cluster of Hox genes. Each gene will be responsible to correlate their signals or cascade their signals to the target gene further, which has to be expressed and take part in the development of the organism through the differentiation process, and that will be expressed in both time and space during development. So here time and space means, as we know that there will be gastrula stage, gastrula stage, and further stages of the development of the embryo that is going to pass through the different time and with the different space. So here different time means every particular developmental stage is going to have its particular time, and space in the sense it is going to develop or increase the embryonic size during the development with the help of the differentiation and formation of the new tissues for the organs in the growing fetus. So that is what the meaning. Next is the Ilium genes. Function in cytoskeleton remodeling at focal adjacent sites as scaffold for protein complexes and as transcription factors. Ilium superclass of genes originated in the stem lineage of animals, and this expansion is thought to have made a major contribution to the origin of animals' multicellularity. Now here you can see, compared to Hox genes, these are also responsible for the origination of the animal multicellularity. Origin of the animal multicellularity in the sense, the animal multicellularity has to initiate from the embryonic stage itself. It is not going to happen as a bomb explodes, but as per this explanation, the Ilium will be responsible for the origin of multicellularity. So these are going to help even in the during the differentiation process, that is during the patterning of the body or the structure, patterning of the structures or the body structures, however it is. Now here I have taken only few points here because Ilium genes has the vast library to be included, but to make you people to understand that even these are the homeotic genes, I have taken these points. Now here you can see that here we are seeing that cytoskeleton remodeling, focal adhesion sites as scaffold for protein complexes, and transcription factors. So here even these are the transcription factors which are going to induce or induct a further targeting signal to a particular cell type to develop or express a gene and differentiate further to that particular tissue and organ. So this is regarding the LIM genes. Now further is PAX genes. Function in embryo segmentation, nervous system development, generation of the eye field, skeletal development, formation of face structures, and many more structures in different species of multicellular organisms. So here it is very clearly giving the explanation that it is responsible for again the embryonic segmentation and nervous system development, generation of the eye field, skeletal development, formation of the face structures, and many more structures in different species of multicellular organism. So this is also a homeotic gene cluster. And POW genes, homeodomain regulators, lead to tissue differentiation in metazoans. Here it is making it very clear, tissue differentiation in metazoans in the homeodomain. POW domain genes have been described in organisms as divergent as Xenoblatis elegans, Drosophila, Xenophus, zebrafish, and human. So here limb gene, LIM gene, PAX gene or POW gene are also the homeotic genes. Now we will see a few slides with some homeotic genes with regarding to the Drosophila itself, and in this we will make some understanding between what are the homeotic genes which we have discussed till now in these slides, and now what we are discussing. In this we can see that in Drosophila there is two cluster of genes. One is antenapedia complex, this one here, and other is the bithorax complex. These are the two clusters which has been observed by inducing the mutation. Here you can see the mutation has been induced at the anterior part, later anterior part, and the middle part in the segments, that is for the sex chrome reduced, then antenapedia. This is the one more cluster, ultrabithorax, abdominal A, abdominal B. So two clusters. These two clusters are located on the single chromosome, hopefully on chromosome number 3 of the Drosophila. Now we will observe the same thing here. In this you will see anterior posterior. Drosophila home C is the sequence of the chromosome number 3, where you will see one cluster here as you have saw in this, that is for the labial deformed sex chromes reduced antenapedia, ultrabithorax, abdominal B. The same thing is here. Now when you are going to observe, this has been corresponded with the ancestral home C, where the same has been corresponded with the human Hox A, B, C, D genes. Now here when you are going to compare this, even Drosophila genes, homeotic genes, or the Hox genes even here. And now why it is called as home C? It is homeotic genes complex which is found on the chromosome number 3. That is why it is called to be as the home C. the similar way it is present in the ancestral also. So here it is the hypothetical sequence. They have not mentioned which ancestor. It may be due to the commonly found gene sequence in the ancestor. So there is the ancestral home C here. So further you can see that it has been corresponded with the human Hox ABCD gene cluster. Now you can see, so here what I am trying to say is that whatever the genes you are studying with regarding with the Drosophila melanogaster homeotic development, so whatever these genes is present, those are named corresponding to the mutation caused. Here, other than this, there are not the direct homeotic genes which are responsible, because the homeodomain is different, which is again falls under the homeobox as the Hox genes and the ParaHox genes. These Hox genes and ParaHox genes are the same in their expressions, so they are together called to be as the HOXL genes. So these HOXL genes will be responsible for this particular homeotic gene expressions during the development. Now here again, as we have discussed this homeotic gene expression, it is the Hox gene cluster. In the next slide, here you can see these are the LIM genes. As we have discussed that LIM genes are responsible for the origination of the multicellularity in the higher grade organisms. Now here you can see that these are the superclass cluster of the LIM genes. Those are again the homeotic in their responses during the development of the organisms. Now here it is the Capsaspora, Salfingoica and Monocyga. These are the organisms. Nemeopsis, Amphimedon, Trichoplax, Nematostella and Drosophila. Here you can see Drosophila, and next the human being. Now with regarding to the each organism, they have mentioned or they have swabbed the boxes with color. Those boxes are the genes which are going to express. Certainly with each organism they are differing, and here with higher grade the expression of this particular LIM homeotic genes are going to increase. For the Drosophila it is higher, for this Nematostella it is higher, and for human being almost all genes are expressing. Even here this cluster is present in the homeobox and expresses through the homeodomain. Now again in this you can see these are the segments, odd even segments of the Drosophila embryo, that is the early embryonic stage, where you can see that the paired mutation has been happened here. That is because of the mutation in the PAX gene. So here these are the PAX genes which are responsible for the induced mutation, where mutation has been caused in the PAX gene. So it has given the difference in the segments. So here hopefully the even segments has got absent because of the mutation. In this you can see that these PAX genes are having these domains to express. So here this is going to have the homeodomain which is going to regulate these PAX genes. And further there is a table which is showing the cluster of the PAX genes, which are 4 clusters with 9 PAX genes. So first cluster is not in the homeodomain, and second cluster is present in the homeodomain, and third cluster is present in the full homeodomain, and fourth is also present in the full homeodomain. So here you can see octopeptide no ectopeptide. So these are the characteristics features or the nature. So you can see these four groups or clusters of the PAX genes. You can understand that these genes will be responsible for the homeotic expressions. We have tried for the POW genes to get some information. So we are not able to get the major part to put across on the slides. So if you are going to prepare separately for Hox genes and Ilium genes, PAX genes and POW genes, we will try to prepare the complete slides so that it will be helpful for you guys to understand the further role of these genes in simplest way, so that it will help you for your further whatever the tests or the examinations. So with this homeotic genes is going to complete. But the overall thing what I want to say is that do not confuse yourself with the homeotic gene explanations which has been given in the tutorials to what the exact picture of the homeotic genes is, because in tutorials hopefully they will give the good information, but as per your examinations they are going to put across the information, but the actual complete information should be like this. I hope I have done a complete explanation about this homeotic genes and how they are in the expression and how the structures are present. And thank you for your kind cooperation to watch these slides, new slides, homeotic genes, and be tuned to the learner's big book. We are going to come with new topics, and we will try to give a authentic information compared to any other tutors. And if you find any flaw in our explanation, please be bold to ask the questions, so we will try to meet your expectations at our level best. And I expect these slides should be shared with your friends and help us with the feedback whether the slides explanation is good, and that I was audible also and I was reachable through a particular good explanation. Thank you, and be tuned to the Learner's Big Book.